Thank you. I'd like to thank everybody for the opportunity for us to come here and meet and talk with you, present some of our work, and also obviously enjoy a few days here in the sunshine. So I know the last day and a half, there's been a lot of discussion about model organisms and how much they have contributed to our basic science understanding. But today I'm going to try to put a human face on some of this. And so I titled my talk, The Tumor Suppressor p53 and the Ultimate Model System, which would be man. So the first question would be, what is p53, if that's the title of our talk? And you'll hear a great deal more about this from Dr. Lozano in the second part of the talk. But just a few highlights. It's a very well-known tumor suppressor gene. It's involved in the DNA response pathway, response to DNA damage, and in trying to limit the long-term effects of that. Importantly for human cancer, it's the most common tumor-specific or somatic mutation observed in human cancers. And this has really been reiterated in the last year or so, where there have been so many tumors where whole genome sequencing has been done. It hasn't been just looking for your most popular gene. And even when all of the tumor's genome has been sequenced, in almost all cases, the single most common mutation observed is that in p53. So it just highlights the important role of achieving a cancer by mutating p53 directly. And lastly, and importantly for this talk, is that heterozygous germline mutations, that means you inherit one mutation and you have one normal wild-type gene in the germline, are associated with a syndrome called Lee-Fraumeni syndrome that we'll talk about today. Now, just to kind of review the p53 mechanisms, a normal cell undergoes division and p53 isn't needed at all. p53 is needed when there's some sort of DNA damage or some sort of limitation on the ability to have a normal mitosis. And at that point, there's an increase in p53 levels, and the cells can go into a G1 arrest, which may end up either repairing and then proceeding onward, or with the cell undergoing apoptosis or autography. And if there is no p53 here, then there's DNA damage, and there's no block here to prevent the cell moving ahead with mitosis. And you end up with cells which may have some degree of DNA damage and do not undergo apoptosis. And if this happens often enough, you accumulate cells with DNA damage, i.e. mutations, which over time can greatly increase the cancer risk. So this led to p53 being labeled the guardian of the genome. Now, so next is, what is Lee-Fraumeni syndrome? And this was the first report in 1969 from investigators Lee and Fraumeni of what they called a familial cancer syndrome involving soft tissue sarcomas, and there are a variety of those, breast cancer, and other neoplasms. And when this was first reported, there was no clue whether this was 1969, remember, whether this was a virus, it was something in the water, something inherited, but whatever it was causing clusters of otherwise relatively rare cancers. So the original identification came through the fact that there were two cousins in a family that had soft tissue sarcoma in children. And when one developed the entire pedigree, there was a lot of breast cancer. And this wasn't just breast cancer in general, this was very young onset breast cancer, mostly in the 20s and early 30s. So we're not dealing with just a common disease that occurs in more than one family member. And then there were some other kind of miscellaneous cancers that the investigators really didn't know whether they were part of a syndrome or chance or whatever. But at any rate, after they wrote up their series of a few families, then what they did was very important. They didn't just drop it and go do something else. They followed these families, and about every 10 years there would be another report from these original families. And what they showed was that from 10-year intervals, new cancers continued to occur. Many of them were soft tissue sarcomas or young onset breast cancer, and they began to demonstrate a more clear pattern of inheritance consistent with autosomal dominant inheritance. So Li-Fraumeni was originally just a descriptive familial cancer syndrome. And to some extent it's still that, because there's still no black and white clear consensus on exactly what the criteria for Li-Fraumeni syndrome should be. But it's autosomal dominantly inherited. Most cases that meet the descriptive criteria are attributable to germline p53 mutations. This confers an increased cancer risk for many cancer sites. And it's really not just children and young adults, although they're obviously critical, and because cancers are rare there, that's how it's often thought of. But there's an increased cancer risk from ages from infancy to the elderly, and I'll show you some examples of that. So don't think cancer that occurs over the age of 25 or over the age of 40 can't be a part of this syndrome. And it's highly penetrant, so that mutation carriers, for the most part, will develop cancer in their lifetime. And this is just a picture of Lee over here, Fred Lee, and Joe Fraumeni from the National Cancer Institute. Now, p53, as I mentioned, is very well known as a somatic mutation in tumors, a tumor-specific mutation. And this is just kind of a laundry list, if you will, of tumors in which p53 mutations are fairly commonly observed. And this is a pretty wide range of tumors. It includes sarcomas, it includes breast cancer, but it includes a lot of common cancers such as lung, pancreas, bladder, prostate, as well as more rare tumors like melanoma or lymphoma, leukemia, or testicular germ cell tumors. So there initially seemed to be sort of a conflict between the frequency of p53 in this wide range of tumors and what was usually thought of as the primary tumors in Li-Fraumeni syndrome. Originally breast and soft tissue sarcomas. Then the description sort of widened a little bit, sarcomas under the age of 45, so it wasn't just children, close degree relative with cancer at a young age under 45, and other cancers in the family that were within the same bloodline or consistent with inherited predisposition. More recently, I think has become fairly universally agreed that there are certain core tumors. That does not mean they are the limit, but that there are core tumors that are really highly frequently associated with it. And these would be, as you might imagine, sarcomas, bone and soft tissue, breast cancer at a young age, brain tumors, and a tumor that's otherwise quite rare in the general population, tumors of the adrenal cortex, and they can be malignant or benign. It's very difficult to distinguish unless you have metastatic disease. So these are now sort of considered the core. Other tumors are allowed in, but it's usually thought that you ought to have at least one of these, and probably one of the more rare ones. Breast cancer is a little too common to use as an index of suspicion by itself. But this is not showing up very well, but it's a family we studied a number of years ago, and the scheme here is that these white-colored individuals had the core type of tumors at the ages under 45 that would get your attention for this syndrome. And you can see there are a lot of them, so if they're relatively rare, either rare by tumor type or rare by age, then this family might get your attention. But it doesn't really seem consistent with an autosomal dominant transmission. There are a lot of people skipping generations and so forth. But those who are shown here that are kind of lightly shaded, they carry the exact same mutation. They also had cancer, but they had the common cancers. They had lung cancer, they had prostate cancer, they had colon cancer, they had pancreatic cancer. They didn't all have these either very rare or very young cancers. And you can see that there are an awful lot of them, but when you link them all up together, you do see something very consistent with autosomal dominant inheritance. But some of these would never have been recognized as a cancer syndrome on their own, and most people can't give you history that would put together a large pedigree like this. So some of the non-core tumors that occur in clearly significant excess in this syndrome are listed here. And again, it's not meant to be exclusive, but ones that are way beyond what you expect in the general population for the age-matched population include lung, hematopoietic, that'd be leukemia, lymphoma, colon, prostate, ovary, pancreas, other GI, particularly gastric, and melanoma. And when you look at this list, you might think, well, maybe it's just one type of lung cancer or just one type of leukemia, but no. It's a wide variety of lung cancers. It's a wide variety of hematopoietic cancers. So the point I'm trying to make is that there are very few things you can latch onto and say, this is Li-Fraumeni syndrome and this isn't. There's a wide range of manifestations, and it's to be thinking of it, you have to be more inclusive than exclusive to actually identify very many families, but they are not all that rare. So this is a revised criteria, which I don't think is particularly helpful, but it is published and often referred to. And it sticks with the age 46 and a proband with one of the core tumors, those 4 tumor types I mentioned, and somebody else who has another core tumor or multiple primary cancers. But then the newer idea that it brings in, which is very important, and you won't see it in all the websites, but that there are some tumors which are rare in the general population in which the frequency of a p53 germline mutation is frequent enough that maybe you should just test all of them. And these would be the childhood tumors such as adrenocortical tumors. Most of these are childhood, although they do occur in young adults. Choroid plexus tumors, which are, again, generally occurring in young children. Embryonal rhabdomyosarcomas. BRCA negative breast cancers under age 30. So these are, again, relatively rare. And importantly, individuals who have multiple primary cancers, at least one of which is one of these core tumors. In the original families that Li and Fraumeni described, when they updated the families over the 10-year intervals, one of the interesting things was that those who survived their first cancer were often seen to develop second or third cancers. And while that can occur in the general population, it doesn't occur very often, and it's much more frequent in these patients. Now, problems with applying some of the criteria from the previous slide are that you notice that long list at the bottom was all rare tumor types, and they're tumor names that most people are not going to know if it's not their immediate family. So getting it right, if you're somebody just taking a family history, is not easy. And it's often misreported, and so getting the kind of information that that last slide would require you to have just doesn't happen, at least in our traditional way of collecting family history information. Secondly, pathology classifications have not been set in stone. Over the last even 20 years, a number of ways of grouping tumors and classifying them has changed. So if you were dealing with a family history in a generation from the past, even if you had pathology reports in front of you, you would probably find that what was written on that pathology report was not the tumor that this last list asked for, but actually it might have been the same kind of tumor. So again, applying those kind of criteria is really tough if you're working in a clinic. And the last one is probably in many ways the most important, and that's you take a family history at a moment in time. Suppose a family comes in with a child age 3 who has a tumor. You take a cancer family history. We know from the first studies of Lee and Framini that those family histories change, and they change fairly rapidly. The general population, they don't change quite as rapidly, but here they do, so it's constantly changing. So again, most of the time in clinical situations, a new patient is admitted and a detailed family history ideally is taken. It is never updated. Almost never is it updated. So if a family history is taken when you're 3 and you come back in at 13 or 25, it's still going to be the same unless there are some changes made in the way we keep medical records and update things so that patterns that are slow to evolve are not just lost. Okay, so breast cancer is the single most common cancer in Lee-Framini syndrome, and this is female breast cancer. To date, no male breast cancers have been observed. It may happen sometime, but here we're talking about one cancer type and yet a population that includes both males and females, and this tumor is more common than all the combined lung cancers or GI cancers or whatever. So there's been a lot of interest in how much does this contribute to female breast cancer. So in the UK a few years ago, there was a population-based survey done. So that means you know your population, you know how many people are there and their ages, and you can calculate exactly how many breast cancers would be expected based on the standard population rates. And in this population, they identified 115 breast cancer cases onset before the age of 31 years. And then they did universal testing of BRCA1, 2, and p53. So this was not based on a cancer family history. It was based on young age of onset and being part of a research study. It turned out that about 7% of the BRCA1 and 2 negative cases had p53 mutations, but not one of them, going back and looking at the cancer family history that had been recorded in the normal sequence of the patient being admitted, none of them had been identified as Lee-Framini syndrome. The cancer family histories were incomplete or not taken or erroneous. So here was a patient who was even in a study, and they didn't get it right for any of those patients. A more important point from this study was because it was population-based, it was over a 10-year, actually almost a 20-year period, they were able to estimate the prevalence of p53 germline mutations in this population. And it came out with a number, covers a fairly wide range, but one between 5 and 10,000 individuals. And that's really the only decent estimate of how common this is. So when you think about rare genetic diseases, this is rare, but you have probably studied in genetics anyway some diseases that are much, much more rare than this. And actually, I think all of this is based on some limited understanding of what Lee-Framini syndrome may really be. Okay, so our approach to studying Lee-Framini syndrome at MD Anderson Cancer Center initially was like everybody else's. We're going to study childhood cancers. Childhood cancers are rare. There's a pretty good survival for many of them. People at MD Anderson especially in these days, we're talking about years quite distant past, starting studies in the immediate years after this and after this. Privacy issues were not such a prominent concern. Patients were very willing to talk to you and give you information and permit you to call all their family members and so forth. So what we did was to set up two separate studies, one studying the families of children who had had the soft tissue sarcomas, the original kind of Li-Fraumeni tumor, and a separate one that studied families of osteosarcoma. For the soft tissue sarcoma cases, they were patients treated during these years. So in 1976, some of these patients had been treated many years ago, and we continued to follow these families. So now we've been following some of these families for more than 50 years. So we've had time to see changes that may occur in families. At any rate, this is the number of kindreds and the total for numbers of families, but because survival was so poor for osteosarcoma, we don't have samples on very many of those original patients. The initial analysis suggested that, that there were a small number of families overall that had way too much cancer to expect by chance, and that it suggested an inherited pattern. Following that, we collected samples on all of the probands who were at that point in time living. And in the 1990s, we then looked for the occurrence of p53 mutations. What we found was that about 10% in each sample set of the survivors, and these are survivors of childhood cancer, about 10% carried a germline p53 mutation. So that's a pretty high percentage of children to carry one of these mutations, and higher than is observed if you look at the data when they come into the clinic and you go based on one of these family cancer pattern recommendations. We also found a few that had a Li-Fraumeni-like pattern of cancer in the families, but were negative for p53. Now, did we miss it? For various reasons, we only looked at the, the coding regions. Did we miss it, or does it represent evidence for other genes that may give rise to these familial patterns? And I don't know the answer to that at this point. So now the next data I'm going to show you are based on a resource, a sort of population resource of Li-Fraumeni syndrome or p53 germline mutation. I'm going to use this two interchangeably, and show you how we've tried to characterize what it means to have a p53 mutation. We have the 18 extended kindreds from those childhood cohort studies I just described to you, where we studied every patient who had come in during that time in a systematic way, and we have now followed those families for many years. We have an additional more than 90 families that have been referred to us through genetic counselors, either within our institution or from outside. So we have a total of well over 100 kindreds, over 300 mutation carriers, and in addition we have over 500 control kindreds from these childhood cohort studies where we collected the data long ago. We have followed them over time, so we can use these in the control families to look for different ages, different tumor types, and so forth, with the same format of data collection. So what do we know about p53 and where mutations are most likely to occur? This is from the cover of Science back in the early 1990s, and this was sort of part of the crystalline structure of the protein. And what this picture shows are the amino acids that were most commonly altered in tumors. These were somatic mutations, and these are amino acids that generally have to do with either the protein binding to DNA or sort of the maintenance of the infrastructure, the conformation of the protein. So there were clearly certain regions that were much, much, much more likely to undergo mutation in tumors than others. When we look at the mutations in our study kindreds, like somatic tumors, most mutations are missense. This is about 65% of the mutations were missense, meaning it's just an amino acid substitution in the protein most often, but it means it's a DNA base change in the DNA. There's a small percentage that are splice mutations, that are nonsense mutations, frame shift mutations, and then a few where there are large deletions or rearrangements within the gene. This is similar to what's seen in tumors. When we then look at the specific amino acids, what we actually see are the same amino acids with one exception that were shown in the tumors. These four here are among the most common in tumors and also the most common in germline mutations as well. So we're seeing the same pattern of mutations in the germline as in tumors. I don't think we necessarily would have predicted that, but that was the finding. This one is really a red herring. It's based on a founder mutation in Brazil and is rarely seen other than in that population. And the rest of these are, for the most part, in the DNA binding domain. So we asked the question of, is there a difference in cancer risk based on what type of mutation you have? Let me go. And we just separated them by the missense mutations because that's where the majority were. And then since the other ones like splice mutations, nonsense mutations, frame shifts are for the most part predicted to truncate the RNA and not lead to a functional protein, we considered these to be two separate groups. And when we looked at the age of onset of tumor based on the cumulative incidence of tumors that were missense versus truncation, basically it's a wash. There was no big difference. This was something of a surprise initially. And these are two samples of pedigrees from our childhood cancer cohorts. This was not ascertained by any other method. One has the commonly agreed on missense mutation that is considered to have a gain of function, and you'll hear much more about that in the next talk. And the other was a frame shift mutation that, if you draw out the predicted sequence after this base deletion, you would hit a stop codon and is not predicted, therefore, to make a functional protein. So in each case, we have a proband with a childhood sarcoma and multiple primary tumors, siblings with young onset cancers and multiple primary tumors, parents with young onset cancers. So it's hard for me to accept the idea that this is a much worse mutation to have than this one. But I do acknowledge that there are selected missense mutations which are shown to have gain of function, and I think at least in some series have shown earlier ages of onset and potential, therefore, clinical and functional implications. The next thing we asked was whether there was a difference in cancer onset based on gender, because I mentioned breast cancer being so common in young women. And this is based on our control families. Cancer is rare at young ages, and it begins to increase in older ages. From the childhood cancer cohorts, followed in the same way. These are from our p53 mutation families, and these are the data for males, and these are the data from females. And you can see that there's a significant difference in the age of onset here. And for example, the median age of onset in women was early 30s, whereas for males it's significantly later, over 45, approaching 50. Now I think when you realize that all the early studies were focused so much on childhood cancers and young onset cancers, A lot of these cancers may have been missed if you weren't continuing to follow a family, because they're sort of getting out of the range that was considered essential to be in Li-Fraumeni syndrome. And you can see that they're not too different here, but the females begin to really drop when you just get a little over 20, when we see breast cancer being a dominant tumor. And you see this steep slope here, and it doesn't really become that steep in males until you get a little further along. We can look at this a little bit different way, looking at the tumor types by age. So this is age distribution in p53 mutation carriers, and this is in a group that we have studied, and I want to explain that the no cancer group should be ignored, because historically, if someone didn't have cancer and was a young child, the general consensus in genetics historically was if you couldn't do something about it, you didn't test someone. So we didn't test children. So this last marker doesn't mean very much. But the others show that as for children, as originally described, soft tissue sarcomas are very common, osteosarcomas are starting to show up, brain tumors, adrenocortical tumors, and an occasional other tumor. As you look at adolescents, as in the general population, osteosarcoma becomes very common. You still see soft tissue sarcomas. You're beginning to see breast cancer in those just under age 20. You see occasional brain tumors, breast cancer under age 20. You see an occasional brain tumor and adrenal tumor. You're beginning to see a little more variety in the tumor types, more other. As you get into 20 to 30, look at breast cancer go up. Huge increase for breast cancer. And as I mentioned, these are all female breast cancer. So this is the main thing accounting for this female early onset. You still see brain tumors, you still see some sarcomas, occasional adrenal, and a few other types. As we get into 30s and 40s, breast cancer is still predominant, sarcomas are still there, but you're seeing a larger percent of the tumors that are other, not one of the core types of tumors. And when you get over 40, the other tumors become dominant. So the older individuals who get tumors in Li-Fraumeni syndrome, if they're not identified because they're part of a family with younger ones, they're going to be totally missed. So in this group, you still see breast cancer, you still see brain tumors, you still see sarcomas, an occasional adrenal, but by far the most common cancer is other. So this is the reason that I feel like in many areas we have missed the boat because we were so focused on a small number of tumors. Okay, so I mentioned previously, yes?
Was this correlated with lifestyle? It didn't reach that point?
It's very hard. What lifestyle do you look at for someone to have breast cancer at age 20? Sarcomas, we have no knowledge of lifestyle factors except things like exposure to radiation. And children and young adults really haven't had much likelihood of that happening, at least not unless they were treated for another cancer. Adrenocortical tumors, we don't know any of the risk factors really for that, except that it occurs much more in young children. So they're brain tumors in children. We don't have a clue of risk factors for those except occasional genetic syndromes. So there's really not much you can look at. We have looked at lung cancer, and as you might imagine, it's increased if you smoke. Duh. You know, these are young people. We don't have issues of obesity and some of the other risk factors we're now thinking about for human cancers. But at this point, I would have to say there's not. Even when you look within a family, you know, in the U.S. we're scattered all over the place. So you can have one SIB that's in California, another one's in Texas, another one's in New York. And you don't see big differences, even though you might expect there could be lifestyle differences or, you know, different geographic settings or something. So at this point, there is very little evidence for, I'm going to get to that later. So as you get into adulthood, yes, you do see a little more interplay of adult risk factors. But for most of these younger people, we don't know any risk factors, honestly. But when you survive a childhood cancer and you've been treated with radiation or chemotherapy, we do know that both of those can be risk factors for additional cancers. And what this slide shows is the risk of a second cancer in over 500 sarcoma kindreds, including those with germline p53 mutations. Again, now we're looking at our starting point at the diagnosis of the first cancer. So this isn't age anymore, it's the years after the first cancer diagnosis. So you had to have a first cancer, now we're looking at your risk factor for another cancer, a new cancer. And so in our general population, which includes the other childhood sarcoma kindreds, some of whom were treated with radiation, most of whom were treated with radiation and chemotherapy, you definitely see some second tumors occurring over time. And it increases over time and begins to escalate a little bit as you get out to over 30 and 35 years. But look at this curve for the p53 germline mutations. Some of these are almost simultaneous. And since you know most of these are very young people, nobody should be having multiple tumors at young ages. And you see this just go up and up and up and up and up. Again here, median age would be between 10 and 15 years after first cancer, whereas for this group, you never get a very high frequency of second cancers. So this is a real hallmark of at least someone who is at risk for p53 mutation. It can occur in the general population, but particularly when you see a short time to tumors, that's a patient who should be considered at risk for p53. Now, of course, it would be much better if we identified them before. And in theory, in some occasions, there's not an alternative treatment that's going to offer them much survival, but in some cases there is. And then you can use a management of their first cancer that is going to confer the minimum or minimal risk of a new cancer. This shows the role of radiation. So again, same group of individuals. These are individuals who are in the general population. This is the risk of second cancers in those who had no radiation, and this is the risk in those who did have radiation, but these are all wild type. And you can see that over time, over 10 years or so, you began to see some second cancers, and it's a tragedy for anyone, and it's of concern to everyone who is involved in cancer treatment. But there is a much greater risk in this population. These are the mutation carriers, and these are those who do not have radiation. They're just developing second cancers spontaneously. And these are the ones who had radiation and have an extremely short latent period. We usually think about radiation-related cancers being something that occurs 20 years or more later, maybe longer. Maybe if it's a young child, it's a little shorter. These are individuals who are under the age of 20 at their first cancer. But here the median is within 5 to 10 years. If you had radiation, the risk is high even if you don't have radiation, but here it's more in the range of 15 to 20 years. So big, big impact. While there's a high risk of a new cancer overall, it's even higher if you were treated with radiation. So to review, these are the general characteristics of the multiple primary tumors in Li-Fraumeni syndrome. Higher risk if you're younger age at first diagnosis. median latency of maybe 6 to 12 years, which is very different than what you see in general. Many of these are going to be the same core tumors. Sarcomas are often a radiation-related cancer. So if we know radiation can cause them and you're predisposed, you might imagine that radiation would play a role in sarcomas. It can play a role in brain tumors. It can play a role in any number of other tumor sites. We're seeing pancreas cancers and GI cancers in people who had abdominal radiation as young people. Those are not terribly typically thought of as radiation-related cancers, but when you've had a heavy dose of radiation, as is used to treat cancer, you see these tumors coming up within the area of the treated field. And this goes on forever. So if you notice some of those pedigrees I showed you a little earlier, individuals had had 4, 5, 6, 7 cancers. They've continued to survive, which is good news, of course, but a continuing risk. It's not something you grow out of. Okay, I'm going to change the topic a little bit. You'll hear a lot more detail about this in the next presentation. But you asked about risk factors, and you might imagine p53 doesn't operate in a vacuum. And we know a good bit about how it's regulated and factors that may be regulators and could play a role in p53 and its role as quote-unquote guardian of the genome. So MDM2 is a very well-known negative regulator of p53. When p53 goes up with DNA damage, it will have a feedback system which initially turns on MDM2, but then MDM2 will downregulate p53 so that these cell cycle arrest or apoptosis doesn't just go on forever. So Arnie Levine some years ago identified a variant in the promoter region of MDM2 which he calls SNP309, and was interested in the impact that a rare variant in this negative regulator might have on cancer risk. And in this situation, the T allele is the common allele, and the G allele is this variant that he identified. And we're not talking now about one of these very rare variants that that you only see in less than 0.05% of the population. This is actually fairly common. So a small percentage will be homozygous, and quite a significant percentage will carry at least one G allele. So even though we had a small population, it seemed reasonable to see what the impact of this variant might be. Now, the hypothesis was that if p53 is a negative regulator, something that changes the amount of MDM2, might affect the p53 available to perform, as I said, its role in DNA response. So what was found was, based on basic science studies in the laboratory initially, was that the SNP309 increased transcription factor binding and increased transcriptionness of MDM2, which therefore attenuated the available p53. And the hypothesis was then that this might lead to earlier onset cancer. And indeed, initially in looking at a population of sarcoma patients, not p53 mutation carriers, there was an earlier age of onset for those with this SNP309 than in the wild type for this SNP. So then our question was, what did this do to p53 germline mutation carriers who already only have one wild type p53? What impact would it have on them? And this shows both our population group and our mutation carriers. And so this is age versus cancer-free survival. So everybody's cancer-free at birth, and this is the cancer occurrence by age. So the general population, we see the slightly earlier onset, and this is all cancers, in general population with the SNP 309 as compared to mutation carriers. Excuse me, these are all wild type, I'm sorry. These are all general population. SNP 309 wild type for that SNP. Now we're going to look at males and females. These are p53 mutation carriers with the wild type T allele, and these are the male mutation carriers who carry the SNP 309, and the same for female. So you can see, number one, the same difference between males and females that I showed you before, but you can also see the impact of the SNP 309, and it does have a bigger impact or shift for females than for males. So there are likely a lot of other genetic variants that may have an impact on p53 and cancer risk in Li-Fraumeni syndrome that may not have been identified. This is an example that can be clearly demonstrated both in the basic science lab as well as by looking at tumors in patient populations. But I think it's a reminder that we focus on a gene and think it does everything, and then we begin to unravel some of the factors, both genetic and otherwise, that may affect the cancer incidence. And here again, we can look at the same thing, the p53 mutation carriers and the multiple primary tumors by the SNP 309. So in those who have the wild type T allele, most of those with cancer just develop one cancer. As we have one G allele, and more so with two G alleles, we see more and more and more with multiple primary tumors. And this is perhaps even more dramatic by looking at these same two pedigrees you've seen before, and they're labeled a little bit differently this time. Mutation carriers have the red spot in the middle, and cancer individuals are shaded in black, but a yellow here would mean that they're p53 wild type. So if we look at them with respect to the SNP 309, we find that of those that we had samples and could measure with young onset cancers, they are mostly of the GG or the GT genotype. Unfortunately, we don't have samples on everybody throughout, because they don't always happen to be living at the time you're interested in collecting samples. But this certainly shows kind of the association with this G allele. Okay, another factor, and this gets back to my story about evolution of cancer family over time. And I think this is really an important finding from clinical perspective, and also potentially for investigation in terms of the biology. This is one of our childhood cohort, just randomly picked up families. Patient had a sarcoma at the age of 1, and at that time he's from a pretty large family. I'm not even showing you the mother's side of the family because it's big too, but it turns out to be irrelevant. No way to know that at the time. But it's a big family, and there's no cancer that has occurred in the family when he presents. The next thing that happens is his parents have another child who isn't shown here, I'll show her in a minute, who also had a sarcoma at the age of 1, and that absolutely got our attention. But this was long before p53 was identified, so we just sort of watched them. Well, as we watched them from 1973 to 2004, this is what we observed. The original proband and his sister both had p53 mutations. All of these individuals related to the father who have the red dot were cancer-free when these two kids came in, and yet they all carried the mutation, and they have all developed cancer, and at this point they have all died. We have in this pedigree a phenocopy, just to show that other people can develop childhood or young adult cancers and die without having a p53 mutation. But an important finding that sort of fascinated us was that in this generation, the mutation carrier was a long-time smoker, 45 plus years of smoking, and died of lung cancer. Initially, we thought that was perhaps not even relevant, but we could later show, based on mutations in the same mutation in relatives of his, that he must have carried the mutation. All of these developed their cancers in 30s and 40s. All of the mutation carriers in this generation developed their cancers in children. Now, this could be anecdotal. I will totally answer that, admit that. But it seemed like we were seeing the same thing in other families. So we tried to look at our group of families at the time. And the way we decided to look at it was we had an older generation and a younger generation. Now, if this was just some change in the environment, change in dietary habits, lack of exercise, something that would affect a large population, then if there was going to be a shift in age at onset of cancer from one generation to the next, even though there might be, then you would expect to see it whether you were a mutation carrier from one generation to the next, or whether you were wild type in one generation to the next. In other words, if it was an important environmental factor for cancer, it shouldn't just affect mutation carriers, it should affect the generation differences for wild type individuals as well. So we looked at that, and we looked at it for males and females because, again, I showed you the difference in cancer incidence. And when we looked at non-carrier men, and you can barely tell, but there are two lines here. One is labeled O and the other is labeled Y for old and young. It's not a very scientific term, but that was how the generations for many different families were lumped together, and older and younger. And there's basically no difference. When you look at the mutation carrier men, there's a big difference. With the younger generation showing a change in age of onset that's different from the older generation. And same for females. No real change between the older and younger generation of females, but a huge change, even bigger change for women who were mutation carriers. Now, curiously, a similar thing to this, not this, was seen by Mary Claire King and group in looking at BRCA mutation carriers for breast cancer. And they attributed it to diet and exercise. But given that we don't see it in patients of the same generation who don't carry the mutation, excuse me, it's hard for me to attribute it to something quite that straightforward and simple. Excuse me. So we felt that there was something biologic, and it's a problem that we have not solved. There has been some evidence for higher number of copy number variants or shorter telomeres, but it's not been consistent from one study to the next, so I'm not sure that that can explain. Now, so again, to summarize, risk modifiers for p53, Lepra-Mini syndrome, gender, female being at greater risk, mutation type and site, perhaps some of the gain of function missense mutations may have a higher cancer risk. The MDM2 SNP309 clearly has an impact. Exposure, I didn't show you smoking data, but I hope you will accept that. Radiation data, and we don't have as much data, but cytotoxic therapy, treatment, for example, with chemotherapy, and then we know from the general population increases risk of myelodysplastic syndrome and acute myelogenous leukemia, and we have some evidence that it increases this even more so in p53 mutation carriers and the generation effect. So I wanted to show you some families and some of the funny things that come up when you're studying genetics in families. So this was one of our childhood cancer cohorts. He had osteosarcoma at 15. And at the time, at the time we initially studied the family, it was long before p53 had been identified as the underlying genetic predisposition. And we were collecting samples with an eye towards doing linkage analysis. And we knew there were a bunch of cancers in between in this part of the family, and we knew there was a child down here that had a brain tumor. So we thought these two people were potential manifestations of the same genetic change, whatever that was. And if we could collect all these samples and track markers through them, we might be able to identify the gene. So we collected all these samples, and as things sometimes go, by chance, this was the patient who was initially analyzed, the sample that was initially analyzed in the lab. And a p53 mutation was identified. I have to fast forward because we just had markers on these and we didn't find anything terribly consistent. But at a later point in time, p53 was looked at and we found that this patient had a p53 mutation. So then we go back to our proband, assuming that that person will also have a p53 mutation, but much to our surprise, he did not. Again, the red circle in the center is a mutation carrier. The yellow circle means they're not. And dark in the bigger circular square means cancer. So this part of the family through which we ascertained this patient has nothing to do with the p53 mutation here. Well, that was a bit of a surprise because there was very little cancer in this family. And so we studied this person's parents and found that this lady at 58 carried a mutation, cancer-free. We found the other child, who's now an adult, carried a mutation. And sort of for fun, we studied this woman's mother, who was living and well at 88, and found that she carried the mutation. And most recently, but not yet added to this slide, this 90-year-old who had prostate cancer also carries the same mutation. So we've got a number of unaffected mutation carriers here. This child would clearly have just looked like a sporadic, random brain tumor with no other cancer in her immediate family that's remarkable, cancers at rather common ages. And it opens up the idea that if you don't look for this sort of thing, you may have mutation carriers in their 60s, 70s, 80s, even 90s, who don't show the usual severe manifestations. So when you go online and it says if you carry a p53 mutation, everyone will have cancer by the age of 50 or 60 or even 80, it simply isn't true. But in general, if you're in a genetics clinic, you don't test everybody else. You don't go testing these people because they don't meet any of the criteria. You would never have tested this child because it didn't meet any of the criteria. So I'm trying to make the point that there's a lot more here that we have yet to fully characterize. And so maybe there are resistance factors that have not yet been identified. And this is another situation that commonly comes up, where you have a person who does have a young onset cancer that would at least be suggestive of p53. Should you test them if they're negative for BRCA? Probably yes, but there's some cancer in the family, but nothing terribly striking. And so one would assume, or in many cases one would assume, that these individuals, if they did test positive, might be new mutations. And new mutations definitely do occur. We see them in maybe 10 or 15% of cases. We can actually confirm that it's a new mutation. Sometimes we can confirm that it arose on the paternal or the maternal chromosome, but we can confirm that it was not in either parent. It is here. The parents are the correct parents, and confirm that it's a new mutation. When you go ahead and study the parents to actually ask that question, in this case, for example, this lady at 51 carried the mutation. Haven't had a chance to study other family members at this point. And in this case, we confirm that the father and sibling also carry the mutation. So it's really hard, unless you're testing other family members, to know when you have a new mutation versus when you have perhaps lack of penetrance at that moment in time. Again, why following families has been so, over time, has been so informative. So it's really the question, when you see something like this, you may not always have all the other branches. Is it a sporadic tumor, just chance? Is it a new mutation, or is it non-penetrance? And of course, if you took all the 3-year-olds with a soft tissue sarcoma, the majority are going to be sporadic. Most are not p53 or any other known mutation. But if you ask then whether it's a new mutation or non-penetrance, at least in our data, it's more often non-penetrance. It can be the male, it can be the father or the mother, but in most cases, it's actually non-penetrance. Now, Li-Fraumeni syndrome and germline p53 mutations may be known in basic science and medical school basic science classes. It is not very well known in the general population, even in terms of practicing oncologists and so forth. So I was very surprised when this paper got published in 2011. There was an unusual patient who had had breast cancer at 32, treated with radiation and chemotherapy. 39, she developed ovarian cancer and treated with chemotherapy several cycles. She did not have a BRCA1 or 2 mutation. And then as time went on, she, a few years later, developed a therapy-related acute myelogenous leukemia. They thought this was so striking that they did whole genome sequencing of the normal tissue from this lady to see if they could identify some genetic susceptible factor, thinking they might identify a new gene. Well, so after going through all of this, what they discovered was a p53 mutation. Never thought about, that's the most expensive p53 mutation analysis probably yet. I know not all of you are going to be out somewhere practicing where this would be super important to you, but somehow the thinking about the second cancers and high risk and all that, if you don't have a family history, it just hasn't reached out very far. These are also pedigrees that were not expected to, but turned out to have p53 germline mutations. Tom Walsh and Mary Claire King were studying ovarian cancer, and they put together a panel of genes to look at one time. So we're not going to, what has been happening in the clinic is a patient studied for BRCA1 and 2. Well, if that's negative, then you come back to the geneticist, get those results, and talk about, well, let's test something else. And then it could be, well, let's test for something else. Well, every time the patient goes through the hassle of anxiety about each gene that comes along, finding it's negative, but we can do another one. Each one costs extra money, and it kind of goes on for a while. So what Mary Claire and Tom did was to put together a panel of genes that were known to be involved in breast and ovarian cancer and look for mutations. And p53 was one of the group of about 60 patients that was in their sample. These were ovarian cancer patients, not selected for cancer family history, for young onset, for multiple primary tumors, for anything, just random older age onset ovarian cancer. And these are the 3 that turned out to have a p53 germline mutation. And I show them to you because none of these families suggest anything unique. They've got older onset cancers, yes. Most of those individuals were deceased. Nothing particularly striking to make you think of p53. None of them would have ever been tested for that in the general medical setting, and yet there they are. And one more interesting family. I think I have hopefully emphasized that within a family with a particular mutation, you don't see the same tumor recurring in every person who carries the mutation. You see a breast cancer, a sarcoma, a brain tumor, a GI cancer, a pancreas. It's not as if a specific mutation confers extra high risk for one or another tumor type for the most part. So this was another interesting family to study because there were multiple cases of leukemia, 5 cases of documented leukemia, 3 of them in children. And this was the pedigree. I was working with a collaborator at Texas Children's Hospital, which is next door to MD Anderson, and we agreed that there was no reason to test p53 in this family. Leukemia is associated with p53 in some cases, but certainly not when you've got 5 cases. And there was a lot of concern because these cases are all deceased. This was a new case diagnosed, and he was going to need to be treated with a bone marrow transplant. Now, if there's something genetic in the family, you really don't want to take one of these sibs and transplant another high-risk bone marrow, right? So there was interest in the family in general, and there was also a somewhat compelling question of how do we figure out what's going on here so we get this patient the best possible transplant. So moving genomic sequencing into clinical application. So the plan was that there were samples that from this patient who had been treated at Texas Children's as well, whole exome sequencing of these 2 affecteds, and this person who is from the other part of the family as sort of a control. And then you would look for something that was in common between these 2 that was not present here. So when this was done, there were many variants found initially, but not so many that were shared by these 2 and not by that person. And when they finally sorted through all of the variants that were left, well, I'm sure you know the end of the story, they found a p53 mutation. In this person, and this person, and definitely not in that person. Also, once we knew the p53 mutation, could test these siblings, determined that this child carries a mutation, this one doesn't, and he is now the donor for this person who is doing well. Not quite the end of story. Since this slide was made and this paper was published in December, this lady showed up at MD Anderson with a melanoma at age 20, and she too carries the same mutation. We've also learned from records that this person had ALL, like the younger ones, at age 33, but at age 36, he had developed AML, a new leukemia, from which he died at that point, presumably related to the treatment for his first leukemia. This just sort of gives you the many faces of germline p53 mutations in families and patients. And this one was an esophageal tumor, and we assume most likely he carried the mutation as well. So now, the important thing, if this is all going on, is what do we do about it? You can't just tell people they have a germline p53 mutation and have a nice life. The first things on this slide are things that have at least been recommended to some extent or another over the years, without having a whole lot of impact. For breast cancer, we all have high-risk breast cancer clinics based on BRCA1 and 2. So we can recommend breast self-examination, clinical breast exam regularly, starting at earlier ages than the general population. A lot of people recommend alternating mammography with MRI every 6 months, again beginning at quite early ages, and consider risk-reducing surgery. And so these kinds of things have been implemented, at least in recent years, for known women who carried p53 mutations. Recently, even national guidelines are recommending at least colonoscopy beginning at age somewhere between 25 and 40 for colon tumors. We can always recommend annual dermatology exams to look for melanoma. Doing blood tests for leukemia lymphoma is really not likely to pick up anything unusual, but I think it sort of makes people feel better, like they're doing something. And in some instances, young children have had annual adrenocortical hormone measurements in the urine or blood, or abdominal ultrasound in young children in families where there had been adrenocortical tumors. With the exception of this evaluation in Brazil, which, as I mentioned, is a very unique situation, I don't think these have had very much impact. There's been a game changer in the past year and a half or so, and that's basically using annual, or depending on interval ages at different intervals, what's called rapid whole body or total body MRI. One goal is not to use radiation, not to be doing CT scans every 6 months or something, because radiation doses accumulate. And if you're talking about doing something from childhood through lifetime, that's not something you want to accumulate. There had been a study using CT, and all it did was show that yes, you could detect an asymptomatic tumor, but all you got out of it was lead time bias. The patient still died. So there was a small study done in Canada using these technologies. Annual rapid total body MRI does not yet even have a CPT code where hospitals can bill for it. It's that new. It's been used in some research, it's used to calibrate how good it is, but it hasn't really met the public market yet, commercial market. But it's something that instead of having to do an MRI of every body part, which would take weeks and not be very favorably received, it permits a total body MRI from chin down that can be done, depending on where it is, between 35 or 40 minutes to an hour. And now, it's not as good a resolution as if you did every individual body part, but it's also doable. And then you have to separately do a brain MRI with contrast. But at any rate, this is the protocol that they developed in Toronto. Now, of course, that's not US and not most of our health insurance plans. But anyway, it was set up as a research study. They were dealing with 12 individuals. They couldn't randomize them, as you might imagine, so people who wanted to go through surveillance did, and those who didn't want to go through surveillance didn't. But you had your kind of cases and controls, if you will. And they started these studies on very young infants, under a year of age. If, you know, I mean, if there was that sort of person in the family. That was the recommendation. And they have followed them now from 2004 to 2010. Now, not everybody joined the study at the same time, so these numbers down here are hard to follow, and some dropped out. But the bottom line was that of all the 10 who had surveillance, cancers developed. It's not that they didn't develop cancers, but all of them are still living out here in 2010. Many of them appear to be cured. 1 or 2 are still on treatment. And some of these were small but very aggressive, highly malignant tumors, such as a choroid plexus cancer in a young child. In those who chose not to have surveillance, all of them who have developed tumors, well, 80% of those who developed tumors have died. So this is a small study. The follow-up period was not forever, but it's the first study of any type that has prevention as its goal or early detection as its goal that has had positive results. So now any number of places around the country are setting up this rapid whole body MRI to use as that and brain MRI as the major screening tools for this syndrome. And we hope that this will change some of the opportunities in future for carriers of these mutations. So I want to make one quick mention. There are a few other genetic syndromes that can look like Li-Fraumeni syndrome. These are all, even for the most part, even more rare. There's something called a rhabdoid tumor syndromes, where you have rhabdoid tumors in children. You can take your mismatch repair genes, and we think about that as a dominantly inherited syndrome like Lynch syndrome that increases risk primarily of colon cancer. But what if you had a mutation in both alleles? Could be compound, so that you got a different allele from each parent, or you may have consanguinity and get the same allele. But when you have biallelic inactivation or mutation in mismatch repair genes, it can give you a syndrome of multiple young onset childhood cancers. And often the parent family history does not particularly resemble the Lynch syndrome. Bilateral inactivation of Fanconi genes can give you a variety of different childhood cancers. And Lynch syndrome, which is usually due to a mutation in one, not both alleles of one of these genes, colon cancers, the most common, other endometrial cancer, but brain tumors are not rare, and occasionally you see sarcomas. And of families like this that we have studied that have sarcomas, colon, and brain, about half of them have been Lynch, and about half of them have been Li-Fraumeni syndrome. And then if you have families with BRCA1 and 2, a lot of breast cancer, the occurrence of a sarcoma can make it, because young onset breast cancer is so common in Li-Fraumeni syndrome, it can resemble Li-Fraumeni syndrome, and you simply have to test for both possibilities. So I've told you, I'll just summarize quickly here. Based on studies at MD Anderson anyway, roughly 10% of unselected childhood sarcoma patients had p53 germline mutations. They're at increased risk for most cancer types. Most of the cancers do not actually occur in children. Cancer risk is higher for females than male mutation carriers. Excuse me. High risk of multiple primary tumors. We have this question of anticipation with earlier onset tumors in successive generations or evolution of the cancer family history. And we have a lot of unexpected things that show up. So there are clinical implications. I understand that the medical students that I thought were going to be here are for the most part not here, but perhaps we'll have access to this through tape. Initially, when you told people about this, people didn't want to know because you couldn't really tell them anything very encouraging about what to do about it. So, and there's still some of that attitude. But now I think there are many more reasons to recommend or at least to consider testing. It's still individual decision, but there are more things to make it a productive option. Then there's a question about when to test. Do you really want to test newborns or infants in the first year of life? Many people who want to test just really can't bring themselves to test children at a very early age. Cancers can occur in the first year of life, but that's certainly not the most common age. We need to do a lot of work on patient and physician education, so that all of our patients, when they go back home and they tell their physician what they have and what they need to do, the local physicians, even oncologists, have generally never heard of this. Obviously, general risk reduction behaviors are good. Don't smoke, don't get out in the sun and get sunburned, so forth. But it's probably helpful if you can do more than that. So now the important thing is that there's some important management options. Now there really may be a surveillance that can actually detect tumors at stages at which they can be successfully treated. We can, in some cases, avoid treatments that may be more toxic for these patients than others. That's not always going to be feasible, and you certainly want to survive this tumor before you worry too much about your next cancer. So this has to be put in the context of where a patient is at a given point in time. There are a lot of pharmaceutical efforts to develop treatments that will target mutant p53. Now, it's not because of Li-Fraumeni syndrome, because that's very rare, but you remember most tumors have mutant p53. So if a pharmaceutical group can come up with something that will target, that will in effect block the impact of a mutant p53, you know, that could be their next blockbuster. So there are lots of efforts, but so far it's been pretty resistant to any find. There now are advocacy groups. The NCI held a workshop about 2 years ago with both scientists and families. Most families had never seen anyone else with a p53 mutation outside of their own family. There was this, you know, intense isolation. So now they've gotten organized, they have an organization of their own, they have a website, they have lawyers, doctors, experienced fundraisers, education. There's really a lot going on in this community now. And then lastly, it's because once people know they carry this mutation, if they're young adults, which many of them are, they want to explore various reproductive options to avoid passing this on. And so without testing, there would be no way to proceed with taking advantage of any of these options. So these are all reasons that testing is becoming much more common now than it was 10 years ago. So lastly, I always feel like I have to say thanks to Fred Lee and Joe Fraumeni for the sort of groundbreaking and pioneering work that they did when genetics was not thought to have much to do with cancer at all, and to all the families that have contributed so much to the work. You'll hear from my collaborator, Dr. Lozano, next, and these are other investigators, student postdocs, and counselors from MD Anderson, and Arnie Levine for his idea to look at the MDM2 SNP 309 and look at its impact on cancer risk. So I don't know whether you want to take a break now or have some questions, or wait and have questions after Dr. Lozano has spoken as well. I saw some heads nodding. Break? Okay, sounds like a good plan. And then we can have questions at the end after both talks, or during the break as well.